Hello everyone, I am AAKRITI and I'm currently pursuing second year microbiology from Ferguson College. And for the manuscript presentation, I have chosen the topic Staphylococcus aureus biofilms, recent developments in biofilm dispersal. So let's start. So first we'll see the authors which have presented this presentation in a really good way. So there were two amazing scientists who worked on this. They were Jessica L. Lister and Alexander R. Horswell, in which Jessica L. Lister is a distinguished researcher specializing in microbiology with a focus on bacterial biofilms. Her work extensively covers the molecular and genetic aspects of biofilm formation and dispersal. And Alexander R. Horswell, significant contributions include identifying key regulatory pathways and developing new strategies for combating biofilm-related infections. Now, background of this literature. The manuscript title Staphylococcus aureus biofilms, recent developments in biofilm dispersal by Jessica L. Lister and Alexander R. Horswell addresses a critical aspect of microbiology and infectious disease management. Staphylococcus aureus is a common bacterium that can cause a range of infections from minor skin infections to severe life-threatening conditions. Its ability to form biofilms, which are complex communities of bacteria encased in a protective matrix, significantly contributes to its pathogenicity and resistance to treatment. Now, significance of this research paper. First is the biofilm formation and persistence. Staphylococcus aureus biofilms are known for their resistance to antibiotics and immune system attacks. Understanding the mechanism behind biofilm formation and dispersal is crucial for developing effective treatments. This manuscript dwells into recent advancement in how these biofilms form, maintain themselves, and eventually disperse to spread infections. Then, dispersal mechanism. The paper focuses on dispersal phase of biofilm life cycles, which is a key factor in persistence and spread of infections. Dispersal is a complex process that involves the release of bacterial cells from the biofilm into the surrounding environment where they can cause new infections. Then, the recent developments. The authors reviewed the latest research and technological advancement in the study of biofilm dispersal, including novel techniques for studying biofilm dynamics and new findings related to the genetic and molecular regulation of dispersal. This review helps integrate recent discoveries into a comprehensive understanding of the biofilm biology. Then, introduction of this research paper. So the main topic of this manuscript is by Jessica L. Lister and Alexander R. Horswell explores the critical aspects of biofilm formation and dispersal in Staphylococcus aureus. Biofilms are structured communities of bacteria that adhere to surfaces and are encased in a self-produced extracellular matrix. This topic is particularly significant due to the role of biofilms in chronic infections and their resistance to conventional treatments. Then there were some research questions which we'll see through this entire presentation and also in the research paper which were put forth. First is the what are the key mechanisms and factors involved in the formation and maintenance of Staphylococcus aureus biofilms. Second is how do biofilms transition from a sessile state into a dispersed state, and what are the molecular and environmental factors that regulate the process? And third, what recent advancements have been made in understanding biofilm dispersal, and how do these developments improve our knowledge of biofilm dynamics? Now, the methodology of this experiment. First is the research review and the literature review, which is obviously very crucial to initiate any research. So first is the comprehensive analysis. The authors conducted a thorough review of existing literature on Staphylococcus aureus biofilms, focusing on both classical and recent research findings. This approach helped them compile and synthesize current knowledge about biofilm formation, maintenance, and dispersal. Then another methodology was experimental techniques. First was biofilm assays. The study employed various biofilm assays to observe and measure biofilm formation and dispersal. Techniques such as crystal violet assay were used to quantify biofilm biomass, while microscopy techniques such as confocal laser scanning microscopy provided detailed visual insights into biofilm structure and dynamics. Second was the genetic manipulation. The author used genetic tools to manipulate and analyze key genes and regulatory pathways involved in biofilm formation and dispersal. This included creating gene knockouts or overexpressing certain genes to observe their effects on biofilm behavior. Third one was molecular techniques. Techniques such as quantitative PCR, qPCR, and RNA sequencing were employed to assess gene expression level associated with biofilm formation and dispersal. These methods help identify key molecular regulators and signaling pathways. Then was advanced imaging and analytical methods. First was microscopy because as Staphylococcus aureus are very minute organisms, so there is the indispensable role of microscopy used in this experiment. So high resolution microscopy techniques including electron microscopy and fluorescence microscopy were used to study the detailed structure of biofilms and track changes during dispersal process. Second one was biochemical analysis. The author used biochemical assays to analyze the composition of the extracellular matrix and identify key components that contribute to biofilm stability and dispersal. Another was methodology is in vivo models and data integration and analysis. So animal model, in some cases, the study included in vivo experiments using animal models to observe biofilm formation and dispersal in a living organism. These models provided insights into the physiological relevance and behavior of biofilms in a more complex environment. Then data integration and analysis, which is very important because if we are conducting a series of experiments, so we need to have a track of the data and analysis of the same. So the study integrated data from various experimental approaches to build a comprehensive understanding of biofilm dynamics. Statistical analysis and data modeling were used to interpret the results and identify significant patterns and trends. Then, from this example, we can see the model of Staphylococcus aureus biofilm growth cycle. In summary, upon coming into contact with the surface, planktonic cells attach to the surface associated proteins. Following attachment, cell divide and begin production of the extracellular matrix, which leads to the formation of a microcolony, as we can see from the diagram as well. As cell division continues, we can see the biomass accumulates and mature biofilm is formed. Environmental signals within the biofilm trigger the activation of dispersal mechanism, and upon dispersal, cell re-enter a planktonic growth state and can seed new insights for biofilm formation. So this is, we can see how biofilm formation and how the division occurs in this state. Then second is biofilm treatment. So treatment of Staphylococcus aureus biofilm. Antibiotic exposure will kill susceptible planktonic cells and metabolically active cells near the surface of the biofilm. However, persister cells and metabolically dormant cells within the biofilm survive and remain protected from immune defenses by the biofilm matrix. Treatment with dispersal agent increases the effectiveness of antibiotic penetration and promote clearance. Antibiotic sensitive cells within the biofilm are exposed and killed after degeneration of the matrix, and the antibiotic tolerant cells such as persisters survive and are susceptible to the immune system. Then after this diagram, we'll see the new instruments and techniques and instrumentation and techniques which were used in this research. First one was advanced microscopy technique. As I told you, we can't see them through our naked eye, so we need microscopy techniques. So first one which was employed was confocal laser scanning microscopy, CLSM. This technique was used to visualize and analyze the three-dimensional structure of biofilms in high resolution. CLSM allows for detailed imaging of biofilm layers and the extracellular matrix, providing insights into biofilm architecture and spatial organization. Then second, so this is the diagram of CSLM, which is very advanced and very beneficial for this kind of research. Then second one was super resolution microscopy, techniques such as STORM, stochastic optical reconstruction microscopy or PALM, photoactivated localization microscopy were employed to achieve higher spatial resolution than conventional microscopy. So other one was genomic and transcriptional approaches. So there was RNA sequencing. This technique was used to profile the global gene expression changes associated with biofilm formation and dispersion. RNA sequence provide a comprehensive view of the transcriptome, identifying key genes and regulatory networks involved in biofilm dynamics. CRISPR Cas9 gene editing. The study employed CRISPR technology for precise genetic modification in Staphylococcus aureus. This approach enabled the authors to investigate the role of specific genes in biofilm formation and dispersal by knocking out or modifying their function. Another one was in situ hybridization. This technique used to detect and visualize specific nucleic acid sequences within biofilm. In situ hybridization helps identifying gene expression patterns and spatial uh, occurrences. Then there was matrix laser desorption ionization mass spectrometry MALDI-TOF-MS. So this was used for analyzing the biochemical composition of the extracellular matrix. This method helps to identify and characterize proteins. Then another one was in vivo fluorescence imaging. This technique was used to observe biofilm formation and dispersal within living animal models. So this used living animal models, fluorescently labeled bacteria and biofilm components enable tracking and imaging of biofilm dynamics in real time. And the last one was microfluidic chamber. The authors utilized microfluidic devices to study biofilm formation and dispersal in a controlled miniature environment. These devices stimulate physiological conditions and allow for real-time monitoring of biofilm behavior under various flow conditions. Now, conclusion of this amazing experiment. So there was complex biofilm architecture. Staphylococcus aureus forms highly structured biofilms with a complex extracellular matrix that plays a crucial role in the biofilm stability and resistance to external threats. The matrix provides both physical protection and a stable environment for bacterial communities. Another is advancement in techniques, as we saw the role of microscope. So recent advancements in microscopy, high throughput screening and genomic techniques have significantly enhanced the understanding of biofilm dynamics. Techniques such as confocal laser scanning microscopy, RNA sequencing, and CRISPR gene editing have provided new insights into biofilm formation and dispersal, revealing novel targets for therapeutic intervention. Mechanism of dispersal. The study identifies and characterizes the molecular and genetic mechanisms that regulate biofilm dispersal, key signaling pathways, and specific genes involved in the transition from a sessile to a planktonic state have been elucidated. These findings highlight the dynamic nature of biofilm behavior and the factors influencing bacterial detachment and spread. Another was biochemical composition. The study reveals detailed information about the biochemical composition of the biofilm matrix, including the identification of key polysaccharides and proteins. These components are crucial for biofilm stability and offer potential targets for disrupting biofilm formation and improving treatment outcomes. So there are some learnings that I got from reading and preparing presentation for this manuscript. So I will share some some of my learnings which I got from this manuscript. First one was complexity of biofilm architecture. So this manuscript highlighted the sophisticated and highly organized structure of Staphylococcus aureus biofilms. The extracellular matrix not only provides physical protection but also contributes to stability and resistance to treatments. And that's what I learned, that understanding this complexity underscores the challenges of eradicating biofilms and emphasizes the need for targeted approaches to disrupt their structure. Then, advancement in research techniques, the use of advanced technology such as confocal laser scanning, microscopy, RNA sequencing, and CRISPR has significantly enhanced the abilities to study biofilms in detail. And what I learned, that these innovative techniques provide deeper insights into biofilm dynamics and allow for more precise manipulation and observation of biofilm behavior, paving the way for new research and therapeutic abilities. And another one was biochemical composition, the identification and characterization of matrix components such as polysaccharide, proteins, offer a better understanding of the biochemical basis of biofilm stability. And from this, I learned that information can be leveraged to design interventions that specifically target and disrupt the biofilm matrix, improving treatment options for chronic infections. So there are some future research plans for me, that building on the findings from the manuscript, Staphylococcus aureus biofilms, recent developments in biofilm dispersion, several avenues for research could further enhance the understanding of biofilm dynamics and improve therapeutic strategies. So first of all, to investigate biofilm interactions with host immune system. So we can use animal models as it was used, but I think the role of animal model was not that much pronounced in this experiment. So we can use animal models to study how Staphylococcus aureus biofilms interact with host immune system, examine how biofilms evade immune responses, and how immune cell influence biofilm formation and dispersal, to understand the interplay between biofilms and host defenses, which could lead to novel strategies for enhancing immune system and responses against biofilm related infections. And then to investigate biofilm formation in different environments, to study how Staphylococcus aureus biofilm forms and behave in various environmental conditions such as different pH levels, temperatures, or surface time. As we know that Staphylococcus aureus is a thermophilic organism, so it can survive in higher temperature ranges. So we should also see how it acts in different pH levels, different temperature ranges, and how it acts on different microfloras. So to understand how environmental factors influence biofilm formation and dispersal, which could inform strategies for controlling biofilm. So, this is all for my presentation. I hope you find this engaging, as I also enjoyed thoroughly preparing this presentation, and I also hope that you liked my presentation. So, thank you for listening and being a gentle observer. Thank you.